Hello, my name is Dr. Ruth Ballard, and I'm going to be your lab instructor in Bio 184 General Genetics this semester. I'm going to be creating a series of short instructional videos on various techniques that we're going to use in the lab. And I'm hoping that these will be very helpful because not only can you watch them before coming to class so that you're a little bit more prepared, but also you can watch these videos over and over again as a reminder of how to do a particular technique. So that's very helpful because you can look back in the future and make sure that you're still doing things correctly. So today's instructional video is going to be about micropipetting. And there's very few things in the lab when you're in molecular biology or genetics or forensics, microbiology, then micropipetting properly. We use very small volumes, ranging from about a microliter all the way up to a mil. And those are very small volumes, so if you make a mistake in your micropipetting, it can really have a really negative downstream effect on the results that you're getting in the lab. So it's not difficult to pipette properly, but there is a little bit of techniquiness to it, and that's what I'm going to go over with you in this video. So to start with, I want you to notice that I have a lab coat on, I have on gloves, and I also have a piece of bench paper down. And what we're trying to do is to create a barrier between ourselves and the experiment. That's for two different reasons. The first reason is you don't want yourself to be contaminated from whatever you might be working with. It might be biohazardous material, living cells, for example, or it might be chemical hazards that you don't want to have come onto your body or breathe in. The other reason that you may not think about as much is contaminating your experiment with you. And under a lot of conditions, that wouldn't be good. It might lead to having cells in your tubes that then would grow up in a culture, say, and you would no longer have a pure culture. In forensics, which I also teach, we're super worried about it because if we're working with evidence, if we get our own cells and DNA into that prep, downstream we're going to end up with a mixed profile and weird things like that. So we're very careful about making sure that we don't contaminate. Okay, the other thing I want to show you is the fact that we have several things on the benchtop, and I want to explain what those are. So the first is that I have a tube, I have a beaker of 1.5 mil microcentrifuge tubes, which is probably the most common size of tube that you're going to use here in the lab. And I want you to notice too that it's covered with aluminum, so an aluminum lid, and the aluminum lid has a piece of tape on it. Now, initially that will be white, but when this beaker of tubes goes into the autoclave to be sterilized, and the autoclave is just a big oven that sterilizes either instruments, tubes, or things like that at high temperature and pressure, what will happen is that black stripes will appear across the tape. And so when you pick up a beaker of tubes, you can look right away at the lid to see if they're sterile. Okay? So if you see the brown stripes, they're sterile, and those are the ones under most conditions you're going to want to be using. This is true also of your micropipette tips. So these are micropipette tips that are yellow that you'll be using in the lab, and those fit the two smaller pipettes that you'll be using. And then we also have these larger micropipette tips for the largest of our micropipettes. So again, you could see that both of those have been sterilized. So we have three different micropipettes that we're going to use in genetics. The largest is the P1000, and it's got a pretty good sized barrel on it, you can see there, and that's the one that fits those large blue tips. And then you'll also have a P200, which micropipettes between 20 microliters and 200 microliters, and this P1000 micropipettes between 200 microliters and 1,000 microliters or a mil. And then we have the smallest of them, which is a P20, and that accurately delivers between 2 microliters and 20 microliters. So you have to be looking carefully at which micropipette you're pulling up to make sure you're pulling the right one up for the volume that you have. That's not usually an issue with the P1000 because it uses the blue tips and those other tips will not fit on it. So it's hard to make a mistake with that one. But the two smaller ones use the same tip. So it's a very common error, say, to pick up a P200, and what you're trying to do is to maybe do 8 microliters. And that particular micropipette is not the one to deliver that. You need the smaller one. So you kind of have to keep an eye on it, especially when you're starting, to make sure you're getting the right micropipette that you need for the volume that you want to deliver. Okay. I also have a tube rack, and in the tube rack I have a tube, and this tube has water in it. So what I'm going to do in this demo is I'm going to deliver water from this tube into a sterile tube. Before I do that, I want to show you a couple of techniques that you can use to get tubes out of your beaker without contaminating the rest of the tubes in the beaker. And the best way to do this, well, there's actually 2 ways, and I don't think either one is necessarily better than the others, but I'm going to show you both. The first one is to use the inside, the sterile inside surface of this aluminum foil as a basket to get your tube. So you can just knock out a few of them. And then I usually just put them down on my bench top, which is clean, and then put the lid back on the beaker. Then I can just close these down and put them in my tube rack, and then those are the ones that I could pipette into. So that's one method. Another method you can use is you can take off the aluminum lid and then just kind of knock them, not out, but toward the front, and just reach in and pull one. And you haven't touched any of the other tubes, and then you can close it down. The second technique, it's usually easier to get just the number of tubes that you want, whereas with the first method, when you're knocking them out into the lid, you tend to get not exactly the number you want. So you might get more than you need, is what typically happens to me anyway, and so I end up having extra tubes that I have out. But anyway, either one of those techniques is perfectly fine. All right. So now I want to demonstrate proper micropipetting technique, and I'm going to use the P200, which again goes between 20 microliters and 200 microliters, and it uses these yellow tips. So the first thing that I'm going to do is I'm going to get one of the tips, and I'm going to press down pretty hard so that I make sure that I've got a good seal between the tip and the top of the, or the bottom of the barrel, physical bottom of the barrel of the micropipette. Then I'm going to open the tube of liquid, this water that I want to deliver into the empty tube, and I'm going to open the empty tube as well. Now you'll notice on this micropipette that it has a plunger, and so you're going to want to put either your thumb or your forefinger on the plunger. When you do that and you push down, you'll meet a point of resistance, and you can actually push past that if you put a lot of pressure on there. So we're going to be using both of those different pressures on the plunger as we're doing micropipetting. But when you're beginning, you just want to push on the plunger to the point of resistance. Then you're going to go into the tube that has the water in it, and you're going to go down below the level of where the water line is, and then gently release the pressure. And when you do that, if you do it well, you should get exactly the volume that you want inside of your tip. And I don't know if you can see that or not, but it comes almost all the way up, because again, I'm at the top range of this micropipetter, so it's pretty high up in the tip. Okay, now it's going to stay in there, that liquid. There are some liquids that tend to come out, like ethanol, for an example, does that. So when you're pipetting things like that, you want to do it real quick so that you don't lose it. But if it's aqueous, it's going to stay in there. You don't have to panic. And then you'll get the tube that you want to deliver the liquid into. And you'll place the tip of the micropipette, notice I don't have any pressure on it at this point, along the side of the tube, because you can get capillary action to help you pull out the last little bit as you're plunging. And then you push down on the plunger to the point of resistance, and then past that, and then keeping that pressure on, pull out, and then you can let go. And that should have then delivered exactly 200 microliters into that other tube. Okay. You can check the tip to make sure that everything came out. It's always good to visually check what's going on as you're going through the micropipetting procedure. And then there's an expel button right on most of these, and you can just expel into whatever container you're going to be collecting your tips in. We'll have containers for biohazardous waste. So these are living cells that we want to get rid of those tips and tubes correctly. So we'll have a box on your bench that's meant for that. And then in addition to the biohazard, we'll also have containers for chemical waste that need to be discarded in a particular way. And then a lot of the tubes can just go in the trash, and we'll try to be very clear about what you need to do to get rid of your tubes and tips safely. The plastic then will be recycled. So if it goes in the trash, it'll get recycled. Otherwise, it needs to get autoclaved if it's biohazardous so that it doesn't have any cells in it. Or if it's chemical hazard, that has to be dealt with in particular ways depending on what chemical it is. So that's where things are going to go downstream. All right, so I hope this video has been helpful in allowing you to see how to do proper micropipetting. There's additional sort of advanced techniques that you'll be learning in the lab as well. This is the very basic, but things like loading an agarose gel and other things, there's a little bit more technique to it that I'll discuss with you later. But in the meantime, thank you for watching the video. I hope you've done it before you came to class, because that's always helpful. And you can use it whenever you like this semester to remind yourself how to micropipette correctly. Okay, see you soon.